Hello, everyone. This is the lecture for Unit 3, lecture number 2. We're talking about blood. We're going to talk about the compositions of blood and also the process for blood to form blood clots. Okay, I'm going to share my screen with you, which I have the PowerPoint notes, like the ones I posted Canvas for everyone. Okay, so let's start from, okay, so we're talking about the blood, right? There's a little difference in terms of the total amount of blood between male and the female. Male has a little bit more because the higher testosterone concentration. Testosterone, one of the functions for testosterone is to stimulate blood production. In terms of blood, hold on one second, let me do this. 484, 8692. I want to join a meeting with my whiteboard so I can use it. Okay, hold on. Okay, so in terms of components of blood, we have plasma, which is the soluble part. Erythrocytes, they are also called red blood cells or RBC. And leukocytes are white blood cells or WBC, and platelets. We'll talk about each one of those. And this one is showing you in the tube is the whole blood. Okay, we put the whole blood in this test tube, and then we put in a machine called a centrifuge to spin this whole blood. And based on the size and based on the size and the components can travel down to the bottom at a different rate. The heaviest part will travel down to the bottom faster. So in this one, you can see we actually can separate the whole blood into 3 parts. The bottom part is all erythrocytes, okay? And that's RBCs, okay? And then the top part, about 55% of the whole volume, that is the plasma soluble part. And then the middle, like a little belt, thin belt right here, this is the part, it's called the Buffy coat, contains all the white blood cells and platelets. Okay, so if we look at plasma, of course, a lot of water here, and also some electrolytes like minerals, sodium, potassium, etc. Nutrients like glucose, amino acids, like gases like dissolved CO2, dissolved oxygen, and also circulating hormones. And notice here in the plasma, we have about 8% in the plasma will be soluble proteins. These proteins are very important because we know proteins build up our bodies, right? So if we look at different types of proteins we can find in the plasma, number 1 is called albumin, it's a type of protein. Number 2 is called globulins, like a hemoglobin in your blood carrying oxygen. Fibrinogen, we'll talk about that in a minute. And so there are many proteins in the plasma. Let's look at the albumins first. This is a huge protein. Molecular size, molecular weight is really big. And there are many functions. One of them is to carry stuff in the circulation, like this one combined with hormones and help the hormone to stay in circulation longer. And also another very important function for albumins is to keep the blood osmolarity, okay? Maintain that osmolarity. So if albumin's concentration drops, dramatically, and then the blood will become thinner, if you can say that way, because the total concentration will be lower. Or you can think that if albumins are lower in the blood, then the blood will have more water, less proteins. If a lot more water in the blood will be very thin, and then the osmolarity is lower, then the blood will try to get out of the water will try to get out of blood, kind of like a simple diffusion. You know, if you have a lot of water in the blood, then the surrounding areas, then the water will try to get out, move out from a higher water concentration point to the lower concentration point. So in that case, you know, the water will diffuse out from blood, okay? And the real case scenario will be for people with malnutrition. If they don't have enough nutrients in their diets, then their blood albumin level will be very low, and the osmolarity changes, and then start having water diffused out from blood vessels and into abdominal cavities. So if you can imagine those in some areas, you know, they have a lot of like poor kids, don't have enough food, and then they seem to have like a big belly because that's water accumulated there, you know, water diffused out from blood vessels. Okay. Next one is globulin. This is another type of big proteins. I will use hemoglobin to explain this later on. Fibrinogen is a fiber-looking protein. So most of these proteins are made in the liver, okay, except the antibodies, all right? Antibodies are made by lymphocytes, the T cells and B cells. All right, if we look at RBCs, these are the basic facts. Should be pretty simple. You know, you can read from your book. Okay, you can look at these RBCs. They kind of like a donut shape, round, flat. In the middle is a concave. So this is we have this kind of shape is actually have a lot of advantages. This way, because it's flat, you can stagger them together, so you can pack more cells in the blood. Two, it has this biconcave shape, so you can squeeze these cells better. Okay, you can squeeze it and make the cell become longer, so it can pass some small capillaries easier. Okay, if you just look at this round shape, the diameter is about 8 micrometer, which is actually higher or larger than some of the capillaries. So in order for these cells to pass capillaries, it has to be squeezed, okay? So this donut shape actually can help, all right, make this squeezing part easier, all right? And another advantage is that this biconcave shape, if you think about here, all right, I say if there is a hemoglobin right here wants to bind to a oxygen, okay? So if the oxygen is on the surface of this RBC, and if hemoglobin is right here, so the oxygen only need to travel very short distance to meet with this hemoglobin and bind together. How about if the hemoglobin is here? If it's a spherical structure, then the oxygen has to travel very long distance and dissolve inside or get into the RBC and then find the hemoglobin here. But if you have a concave shape, look here, is concave shape. So this hemoglobin, instead of waiting for oxygen to come in this way, it can get oxygen on the surface very close by. So this concave shape is really has a functional impacts in terms of getting oxygen inside of RBC. Okay, there's another protein. Let me see if I can move this to the side. Okay, there's another protein here called the spectrin. Okay, so the spectrin is a protein which can keep the RBC in this concave shape and make the cells flexible, right? Okay, in the RBCs, we have the hemoglobin, that is the globulin protein, which are involved in carrying oxygen. We'll talk about them later. And also we have other enzymes I'll talk about. Okay, so this is the, I don't know, what is, okay. This is the hemoglobin structure. Okay, you can see this hemoglobin protein is made by 4 peptides. See, here is 1, number 2, number 3, number 4. So this one is darker red. This one and this one, they're the same. It's called alpha subunit. And this one, lighter red, beta subunit. So one hemoglobin is made by 2 alpha subunits and 2 beta subunits. Okay, in each of these subunits, in the middle, there's a structure, this yellow part here, the yellow part is called a heme group. Okay, in the heme group, this is where the iron is bind to. You need to have the iron inside the heme group, so this heme group can bind to oxygen. Okay, so oxygen, there's an oxygen binding site in the heme group. Okay. Okay, once the oxygen bind to the hemoglobin, then your blood will turn to brighter red color. If you have oxygen unload from the hemoglobin, then the blood will be darker. So if you look at your veins on the surface of your body, you can see the blood is kind of like a bluish looking because it's more oxygen, it is oxygen poor blood in the veins, in your systemic veins. And the hemoglobin can also carry CO2 and carbon monoxide. But CO2 bind to hemoglobin at different binding sites than oxygen. So carbon dioxide and oxygen, they do not compete for the same binding site. However, carbon monoxide does compete with oxygen for the binding site. So let me give you an example here. If somebody stayed inside the car, had the machine, had the car running, have the car running for a long time and sit inside and fall asleep, and then the emission, you'll have a high concentration of carbon monoxide, and then you can get poisoning from carbon monoxide. And for these patients, this carbon monoxide, normally in the atmosphere, the concentration is very low. So there's no competition with oxygen because oxygen concentration is much, much higher. But in the situation, if you have a higher concentration of carbon monoxide, then this competition will start. So carbon monoxide and oxygen, they're competing for the same binding sites. And once the site is occupied by carbon monoxide, this affinity or this binding strength, it's very, very strong. It's much stronger than this binding site bind to oxygen molecules. So in other words, once the CO binds to the on the hemoglobin, it is hard to get them off. And then once they occupy these binding sites on the hemoglobin, then oxygen cannot get on. So think about it, you do need to have oxygen in your body to help with ATP production. ATP is the energy source that your cells can use. So if you don't have enough oxygen, of course, you don't have enough ATP, and then you die. So carbon monoxide poisoning is very dangerous. So in this case, the patients, they have to be placed in this barrel chamber to give them high concentration of oxygen, at the same time increase the pressure in this chamber, because remember I said the binding is really, really tight. You need to have a higher pressure to push to break this binding, at the same time like 100% of oxygen, to compete with the COs, and then you can have oxygen replace these binding sites on the hemoglobin. All right, so this is the carbon dioxide can travel in the blood in the form of these two ions, hydrogen ions and bicarbonate ions. This is a simple chemical reactions you probably learned already. It is CO2 and water can be converted to the carbonic acid, and the carbonic acid can partially break down into hydrogen ions and bicarbonate ions. And this process is very important once we get to the respiratory system to talk about how the CO2 being transported in your body. This process can, you do need to have an enzyme for these chemical reactions. This enzyme is called carbonic anhydrase. This enzyme you can find in the RBC. So for the lifespan of the RBCs is about 120 days. So your bone marrow will continue to make RBCs, all right? And this RBC production is called erythropoiesis. And the hormone is called erythropoietin. This hormone stimulates erythropoiesis. This hormone is made in the kidneys, all right? And then the function is to stimulate RBC production. All right. This slide shows you the blood cell production. Okay, so we all start from one stem cell, one type of stem cell. It is called a hematopoietic stem cell. And from this one type of cell, it will differentiate to make a myeloid stem cell and a lymphoid stem cell. If it's a lymphoid stem cell, it only develops into lymphocytes, the T cells and B cells. The rest types of blood cells and platelets are all developed from myeloid stem cell. This is the RBC production. So you start from this stem cell right here. Okay, hemocytoplast. is the same as this hematopoietic stem cell. It's the same, okay? So this cell, once it's committed to make RBCs, is going to go through these multiple stepped process and finally to make it an RBC here. The important part is here in this way of developing at some point, the nucleus of the RBC will eject out. Okay, so the mature RBC does not have a nucleus. So this RBC, because it doesn't have a nucleus, so there's no point for cell division, right? There's no genetic information anymore. So the RBC will have lifespan about 120 days and then just get, lyse, and some stuff will be reused and others will be eliminated. Okay, to make RBC, we do need to have all these materials, okay? And important one, you all know, the irons, that is a part of the heme group. Without iron, you cannot make a functional hemoglobin, and that means you cannot carry oxygen. The regulation, remember this hormone we talked about, erythropoietin, okay? That is the major hormone involved in regulation of RBC production. Also, testosterone stimulate RBC production. And hypoxia, that means the low oxygen concentration actually has an effect on RBC production. In terms of breaking down RBCs, if the RBCs are old, they can be broken down in the spleen, and most of the irons can be recycled and can be reused. And the heme group will break down, okay? Once you extract the irons out, and then the heme group can be break down into a material called a bilirubin. The bilirubin has a color, it's kind of like dark brown color, okay? So once you have the bilirubin, then the liver can help, well, I should say the heme group, once you extracted the irons out, and then the liver will help to break down the rest of the heme group to form bilirubin. And then bilirubin can be eliminated from feces or from kidney in urine to form urine and get out. Okay, so many premature babies, they may have jaundice. Jaundice is kind of like, is a condition, have yellow skin. The reason for that is at birth, all the fetal blood needs to be replaced with adult type of blood. There's a difference there. And because there's such a huge amount of blood cell needs to be break down, and you're going to generate a lot of bilirubin. And if the bilirubin get accumulated, then it's going to show the color on the surface, on the skin. Okay, that's called a jaundice. But usually this condition can be reversed, by getting more UV light. The baby's liver will become stronger and stronger and can be fully functional. Then jaundice will disappear. Okay, for RBC disorders, okay, the common ones will be anemia. Okay, there are different types of anemic conditions, and I have these listed in your lab handout, unit 3 lab handout. there's a chart. You need to read your book and fill it in, the answers, okay? If it's too many RBCs, then it's called a polycythemia, too many RBCs. That's not good either, because too many RBCs will make your blood very, very thick. You know, we talk about if the blood becomes thicker, then the blood pressure has to be higher to keep the normal flow, right? Okay, so please read your book and work on the lab handout, the chart for anemia. Okay, so I'm not going to talk about anemia here anymore. And there's one condition, it's called a sickle cell anemia, which is a genetic problem. But this gene for the sickle cell anemia, well, first, sickle cell anemia is the hemoglobin is abnormal hemoglobin. And since the hemoglobins are abnormal, then the RBC, the shape, instead of a donut shape, now they are more like a sickle shape, okay? So these type of the cells, it's much more rigid than the normal RBCs. It's much harder for them to pass small capillaries, so they may get stuck in the capillaries and rupture the capillaries, cause the internal bleeding. That's one problem. Also, sickle cells, because the abnormal hemoglobins, they cannot carry a normal amount of oxygen. That's why it's an anemic condition, okay? This is a genetic problem, and this sickle cell anemia is caused by two recessive genes. So if these two recessive genes meet, together, then this person will have sickle cell anemia. So if you look on this side, okay, this male side, see this trait of genes is A and S. A is normal, S is a sickle cell, the gene which can cause sickle cell anemia. But because you have one normal, one abnormal, the abnormal one is recessive. So this person, we say it is a carrier. It does not have sickle cell anemia, okay? Then the female side, the same kind of genotype here. And the offspring, when you make offsprings, okay? So there are four possibilities, okay? So if this normal gene from male side, that's the sperm, and the normal gene in the egg from the female side, if these two meet to make a offspring, then this child is a normal child, okay? Another possibility is this abnormal gene from the male and a normal gene from the female, then you have a carrier. Still, this child does not have sickle cell anemia, okay? The third one is the normal gene from the father, abnormal gene from the mother. Again, you're making a carrier. So this child does not have sickle cell anemia. But when this abnormal gene from the father combined with the abnormal gene from the mother, then you have two recessive genes together, then this child will have sickle cell anemia. Okay, so this is a genetic problem. All right, so we can skip this one, all right? And then about white blood cells, okay? We can divide white blood cells into granulocytes and agranulocytes, I do have a chart in your lab handout. You need to read your book and work on that chart. And also, please study the histology pictures from your book. Be able to recognize each type of RBCs. So I'm going to skip this part of lecture because this is really straightforward. You just need to read and work on the chart, okay? All right. This is a saying to help you to remember the WBCs from the most popular one to the least popular one in that order, okay? Never let monkeys eat bananas. Bananas for basophils, that's the most unpopular type of RBC. Okay, so we need to focus on this topic today. That is actually I want to spend more time on. This is called hemostasis, or you can say it's the process of stop bleeding. It's called hemostasis. Okay, before we start talking about that process, this is a review. Okay, this is a review. Okay, we say blood cell production always start from a stem cell. Okay, hemocytoplast, and this hemocytoplast will divide or differentiate to form a myeloid stem cell and lymphoid stem cell. Okay, lymphoid stem cell will make lymphocytes, right? And the myeloid stem cells will make RBCs and the rest types of WBCs, plus platelets. Okay, so again, this WBC disorders, you have a chart in your lab handout. Please work on that. Okay, read and work on that. Okay, let's talk a little bit about the platelets. Platelets are not cellular structures, okay? They are fragments of megakaryocytes. Megakaryocytes are cells, type of cells, all right? So there are many functions associated with platelets. It's very important, okay? Inside the platelets, there are chemicals like serotonin, which is a very potent vasoconstrictor, and we have a lot of calcium, ADP, and adenosine diphosphate, okay? And also a growth factor called platelet-derived growth factor, which is necessary for forming or repairing blood vessels. So platelets are very important in terms of stop bleeding. Okay, platelets are made from, okay, again, from this stem cell hemocytoplast, right? And differentiate to become a megakaryocyte. This big cell is called megakaryocytes, or megakaryoblast is young, and the megakaryocytes are the mature one. Once this cell is mature, then it's going to break down to form many little fragments. These fragments are membrane-bound fragments, kind of like a little organelles, right? So it's like a membrane, and inside of a membrane carrying many different materials, like we talked about. Let's go back, like say serotonin here, calcium enzymes, PDGF. Okay, all these are can be found in platelets. So now we're talking about this stop bleeding process. It's called a hemostasis. Hemostasis. okay? And in terms of the process, there are three major steps. The first step is called a vascular spasm. Number two is called a platelet plug formation. And the third one is called a coagulation, or the actual step to form blood clots, coagulation. So even though we say there are three steps, but you cannot say vascular spasms happen first and then platelet plug happens the next. It's not that way. It's pretty much all the three steps take place at the same time. And the vascular spasm and the platelet plug formation, they are probably happening at the same time, and the coagulation takes a little bit longer to have the effects. Okay, so let's talk about vascular spasm. Okay, when the vessel wall is damaged, then it's going to trigger a reaction. Okay, this reaction will it's from releasing of the chemicals from the smooth muscle cells around the vessel wall, and also from the lining cells. So these chemicals will trigger this contraction of the smooth muscle cells around the vessel to make the vessel diameter decrease, okay, constrict the blood vessels to minimize the blood loss, okay? That's called a vascular spasm. The next step, the platelet plug formation. This part is when platelets normally travel in blood, they are inactive. But once there's a damage on the lining of the blood vessel, then the collagen fibers from underlying connective tissue will be exposed to the platelets. Then the platelets can bind to the collagen fibers. This will activate platelets and then make the platelets very sticky. So all these platelets, they're going to clumping together. That is called platelet aggregation, okay? Once they get together, they get more activated and attract more platelets to get to the place to form this plug, you know. It's all like aggregated platelets together, try to stop or minimize the blood loss. The final step is called coagulation. That is actually the form, the process to form the actual clot, okay? So there are two mechanisms. One is called intrinsic, another one is extrinsic mechanisms, but the end effect is the same, okay, to form a clot. Intrinsic definitely is the factors coming within the blood vessel. Extrinsic is the factors involved in chemicals released from the surrounding tissue. Okay, so if you look at this intrinsic on this way, extrinsic on this side, and there are clotting factors or clotting proteins involved. There are 13 different clotting proteins. They're all made in liver, okay? Normally they're in the circulation, they stay inactive, all right? But if something triggers this activation, then you're going to have a cascade kind of activation process. That means one clotting factor activation will lead to another one, and finally cause the clot formation, okay? So you can see the intrinsic pathway. Remember, the factors are coming from within the blood vessel. So here we go, you see this endothelium, that's the lining part, got damaged and exposed collagen fibers, attracts platelets and activate platelets, then activate the clotting factors. So you don't have to remember all these factors, okay? But just have an idea. You can see that a lot of factors involved in this intrinsic pathway, okay? If you compare this one to, on this side, extrinsic pathway, okay? So this one has to be the factors outside of the blood vessels that are involved. So if you have a damage, not only cut the blood vessels, but also cut the surrounding connective tissue, the muscle tissue, then you're going to trigger the extrinsic pathway. So if you look at extrinsic pathway, fewer steps are involved. So extrinsic pathway actually is a faster and a stronger pathway, okay? So once you have the clotting factor 10 being activated right here, both of these pathways will lead to the activation of clotting factor number 10. Okay, once you have this factor activated, then the rest of the steps to form the clots, this part are shared by both intrinsic and extrinsic pathways. Okay, so here we go. Another slide to review, okay? So damage of the tissue, activate platelets, right? And then triggers the coagulation, triggers the blood clotting formation process. And then their intrinsic and extrinsic pathways both are lead to the activation of clotting factor number 10. And then the rest steps are shared by both intrinsic and extrinsic pathways. Okay, this one is showing the actual clots. So when we say there's a blood clot formation, what is a clot? This clot is formed by this mash-looking structure, which is a fibrin protein. It's a fiber-looking proteins. And everything get attracted in this mash structure. This whole thing is called a clot. So clot is not just the proteins. It's not just the blood cells. It is the combination of the fibrin and blood cells and debris. Whatever stuff gets stuck over here together is called a clot. Okay, for clot retraction and repair, please follow your book. Ignore these few slides, just read your book. I think your book talk about this part probably is easier than my slides. So let's just use your book. Understand what is clot retraction and how can vessels be repaired. Okay. Removal of clot factors. This part, just read this. Just read this slide. That's good enough. Okay, disorders of hemostasis. Maybe we just pick up one, like a bleeding disorders, like a thrombocytopenia. This is the problem I have. Okay, so I have very, very low platelets. Normal amount should be 140 to 400. But I only have, how much I have? I have 30,000. Yes, normally should be 140 to 400,000. I have 30,000. Yeah, so if you don't have enough platelets, of course, you're going to have bleeding problems. Remember, we said the platelets are involved in forming blood clots, right? So this can, well, for my condition, they cannot figure out what is the cause for that. So they just say, oh, it's an autoimmune disease. You must have antibodies developed against your own platelets to destroy them. But other factors, like say, if you have liver problems, it can cause this condition as well. This is hemophilia. This is a bleeding disorder due to lacking of one or more of the clotting factors involved in this process. So remember we said the clotting factors, there are 13 of them. And if you miss one, you know, it's all cascade process. If you miss one in the middle, then the rest of the reactions cannot take place. Then you cannot form clots, right? Okay, so forming clots is a good thing, right? In many ways, you know, if you have if you traumatize somewhere, you know, cut your blood vessels, you want to stop bleeding, and that is important. But at the same time, you don't want to form a lot of clots in your blood circulation normally. But do we normally form clots? Yes, we do. Yes, we do. But normally the clots we form in the blood circulation, they are very small. It can be washed away, can be breaking down by active circulation, and then it will not get stuck anywhere, all right? But if you become inactive, the blood circulation is slowing down, and then you have higher risks to have these little tiny clots, you know, aggregated together to form big clots in the blood vessels, and then it can get, you know, stuck somewhere. Yeah, so that is a big problem. Okay. Let's say if you have high blood pressure, and then that can be a factor to increase the risk to have more blood clots formed in your blood circulation. The reason is the higher blood pressure, then you have a higher pressure against the vessel wall, you have higher risks to damage the lining. Also, if you have arterial sclerosis, that's, you know, you have something built up in the blood vessels, like high cholesterol, you know, the bad cholesterols build up inside of the blood vessel. Then the blood flow going through these blood vessels, they're not a smooth flow. They're going to have bumps. If they have bumps, they're going to have turbulence kind of, and then messed up the pressure. So you won't have like an even pressure against the vessel wall. You're going to have spots with more pressure all the time. That will trigger that will damage the lining and trigger the clotting process. Yeah. Okay. All right. That is the today's lecture. But before I go, I want to ask you, you know, if you read your book, which you should, okay, the reading pages should be from page 642 to 661, all right? Read 642 to 661. And also, we were talking about blood, forming blood clots, the coagulation part, okay? So on page 659, on the bottom part, gave you the 3 steps shared by both intrinsic and extrinsic pathways. So you really need to look at that pathway, okay? Make sure you understand the last 3 parts. The factors involved in the intrinsic and extrinsic pathways before the activation of clotting factor 10. I don't want you to memorize any of that, but after the clotting factor 10 activation till the end to form the clots, these 3 steps I do want you to know, okay? You should know this. Alrighty, that's it for today. Thank you.